So yeah, we have presented data, which is a new analysis from CARTITUDE-1 trial by looking at the patients who have achieved minimal residual disease inactivity. So that's what we call MRD. So the way the procedure is performed is that you basically perform aspiration of the bone marrow sample from the patient in the study. And then we use next generation sequencing to detect whether there's any cancer cells left in the bone marrow or not. Because really multiple myeloma is a bone marrow disease. That's where all the B cells originate from. So if you look at this data we presented at ASH, you will see that among the 61 patients who are evaluable for this minimal residual disease inactivity or MRD, we had 92% of the patients who achieved negative MRD, which means you can't really detect the cancer cells in the bone marrow. And also we divide those patients into subgroups who have MRD inactivity for shorter than 6 months, or who have MRD inactivity for 6 to 12 months, or who actually can have very sustainable MRD for longer than 12 months. Now, if you look at the data set, you will see that those patients who can achieve MRD inactivity for longer than 12 months had the best outcome. And then, if you look at the patients who only had fewer than 6 months of MRD inactivity, the progression median survival or duration of response was shorter. So basically, the conclusion here is that the patients who achieved sustained MRD inactivity had deeper responses, and then compared to the patients who did not have MRD inactivity more than 6 months. So that's a good predictor the doctors can use in the treatment of multiple myeloma patients. That's the data we presented for CARTITUDE-1 MRD inactivity analysis. And then we also presented data from a phase 2 trial called CARTITUDE-2, looking at the efficacy of Cilta-Cel among patients who have earlier stage of disease. So we presented data from a cohort B. These are patients who basically had early failures from frontline trials. So either they had a bone marrow stem cell transplant, or if they're not fit for transplant, they received the standard cocktail treatment. But unfortunately, the disease came back or relapsed within 12 months of the treatment. We also presented data from cohort C. So these patients have already been treated with prior therapies targeting BCMA. Either it's a drug called ADC, antibody drug conjugate from GSK, or they've been treated in the clinical trials by a type of drug called bispecific antibodies. So these are really, really difficult to treat patients. And then they came into our trial to see whether they can still respond to Cilta-Cel after being treated with other BCMA targeting therapy. So those are the two data sets we presented for CARTITUDE-2.